Yeah, okay. So, yeah, let me begin by thanking the organizers, uh, Debashish, Prabal, and Ambarish, for this invitation to attend this very nice meeting. And as Shakuntala was saying a couple of days back, that it is really great to be a part of this meeting, like physically present here after like 2 years of confinement. And that's why I thought that I will talk about first passage process in confinement. Okay, so, and yeah, so before I go into discussing the problem, yeah, let me kind of give a brief overview of what are the problems, or in general, what is the kind of problem in biophysics we are interested in. So let me show you a movie. So this is a fantastic example of so many things happening at so many scales, and a beautiful application of example of emergent phenomena. So what you see here is the wing epithelia of a fruit fly at a very early stage of development at like 16 hours after puparium formation, and this wing has been imaged in vivo over a span of 16 hours. And what you see as the fluorescent dots are the proteins at the boundary of the cell, which has been labeled. So one can actually resolve the fluctuations of each cell, or resolve the mechanics of each cell here. And what you see is, as time progresses, this is a time-lapse movie, as time progresses, this whole wing epithelia is dynamically remodeled, and there are a lot of things that are happening. Okay, so there are stresses generated at the level of individual cells, there are cell division, there are cell death, and there are collective interaction between cells. So if you think of this, like you are able to zoom inside a single cell, you will see lot of signaling pathways, lot of activity, which is keeping the cell alive, okay, and lot of proteins created and all. But now, if you come out of the cell and look at, like, zoom out and see many cells, you will see that the collective interaction of all these individual cells which are functional leads to formation of, like, emergence of nice complex pattern. You see the nice formation of structures like veins. You see collective flow from the right side to the left side. And the question is, I think somehow the arrow got messed up, but nevertheless. So the question is how one can think that, like, whatever happening at the level of individual cells leads to, like, formation of complex patterns at large scales. Like, you have, like, different things happening at different scales. So can one, like, understand these things using like principles of physics? And the analogous problem you can think about, like, is from equilibrium physics, where you know that if you have individual, like, entities for which you can write down the equation of motion, say atoms, molecules, or whatever. But now when you put lot of these individual entities together and put interaction between them, you will see emergence of collective order, like you know already, like liquid gas transition, magnetization, and even more exotic properties like superconductivity and all. So here you can think of this as a similar problem, like how collective interaction between individual entities can lead to, like, emergence of like complex patterns. And this is, like, more complex because this system is perpetually out of equilibrium. It is using its own energy to drive itself out of equilibrium. So there are processes which are important at the level of individual cells, which are very fast time scale processes, and there are very long time scale processes which are also important. And we are interested, like, how, like, I mean, in problems related to these different kind of processes, and how we can use principles from statistical mechanics or nonlinear dynamics to understand such things. So this, I wanted to give you an overview. And let me come to the problem which I'm going to discuss today. So I will try to go a little slow. So if you have questions, you can stop me. Okay. So I think during this meeting, there has been several talks on first passage processes, but I thought nevertheless, I will just give you a more general introduction. So what is a first passage process? So you can think of that there is a random variable which is evolving with time. And this variable is not evolving according to deterministic laws. So there are some uncertainties. It's a stochastic variable. And then the question is, when does it variable reach a threshold value for the first time? Okay, and this variable can be anything. It can be like the position of a particle, it can be some chemical concentration, it can be some stock market index or whatnot. And it is given by some equation which has a probabilistic, and it can have also a partial deterministic evolution, but there has to be certain uncertainty. And I mean, this is a schematic, so there is a random walker and there is a target, and the question is, I mean, this random walker follows like a completely uncertain zigzag path, and when does the random walker hit the lamp for the first time? Now, if as an experimentalist you can record all the times like the random walker does this walk for n times, and every time it will be a different time, you will have a distribution at time, and from that you can calculate the probability or the first passage probability density. Okay, so that will tell you how these distributions of the first hitting time looks like, and you can also relate it to something called a survival probability, namely how long this walker survived before it hit the this thing, the lamp. Okay, and it is related to the survival probability through this. So again, so Debashish told me to keep some pedagogy, so I actually kept like a simple example of this. So in the next slide, and this process has several applications in natural sciences, and some of them you have already heard during this meeting. So a simple example you can think of is the diffusion of a particle in a fluid, which is like, I mean, imparting random kicks on the particle, and it has to reach this flag, and whenever it reaches this flag, the game is over. Now the question is, how would you calculate the first passage time? Again, if you are an experimentalist, you can put this thing under the microscope, you can record every time how long it takes, you do this experiment n times, you calculate the histogram. If you know the equation of a random walker, what you can do is you can do a computer experiment. You can write down the equation of motion in the overdamped limit. You know that in the overdamped limit there are no inertial terms, and it is getting these random kicks because of the viscous medium. And this is a purely Markov process. And you can evolve a trajectory. You can evolve in such trajectories. You can calculate the histogram of how long it takes to reach the target. You can also, given that you know this equation, you can ask that what is the probability of finding the particle x at time t, and this equation, the continuum version of this equation is the Fokker-Planck equation or the diffusion equation. Okay, and you can write down the time evolution equation of the probability of finding a particle at position x, and once you have this, you can also solve this equation directly for this geometric scenario where you have a target of some size here. You can think of it, so here I have written the equation for 1D. So you can think like in a 1D axis, you have an absorbing wall at one end and the other end goes to infinity, and the particle is doing like a zigzag motion, and you want to see when does it hit this thing for the first time. And if you can find out, calculate the probability, you can integrate over all space, and that will give you the survival probability. Okay, a more elegant method is you write down something called the backward Fokker-Planck equation. Basically, you write down a time evolution equation for the survival probability. And this can be derived systematically. There are various ways one can derive from this forward Fokker-Planck equation. And once you have that, you can directly solve this and get the same results. So one can show that I'm not going to derive this, but if you have, like in 1D, if you have a 1D box where this particle can move back and forth, and there you have an absorbing wall, then in this case, the survival probability, if it starts from x naught, at long times, it scales as t to the power minus half. I mean, why I write this, you will see in the coming slides. So it follows a power law with a certain exponent. Okay, one can solve this and show. And one can solve this in like higher dimensions also. So there the dimension will come in the exponent, and one can do that. Now the question is, I should remind you, because we are interested in this kind of problem inside the cell, this is a very simplistic assumption that considering the cytoplasm as a very simple viscous liquid. Inside the cell, it will not be that simple. It will be more complicated because you have like a complex environment. You have this network of microtubules, and a simple thing you can visualize here, although it will be even not that simple the way I am putting the schematic, that you can think that, ask the question that how the particle reaches from A to B, for example, here to here. So it can basically a two-step process where it can move along the microtubules by binding to a molecular motor, moves ballistically, and then it can unbind and move diffusively, which allows it to find a direction. And then again, it can unbind until it reaches here. So this will be a more complex process. And of course, the environment is highly crowded, and it is not like a simple fluid. It is viscoelastic typically. So there will be effects of crowding and all. So it will be very complicated. And we are not going to even discuss this. What we'll discuss is a different situation. Say, suppose, let us consider the same walker. Now I consider what happens if I put some boundaries on the walker, that it cannot, it is trapped. Whatever random walk it does, it has to do within some cage. Okay. So now one can solve this, as I will show in the coming slides. So we have put a confinement. Now add a level of additional complexity. We ask that we have this target, now we have several walkers. But it is still a simple scenario that all these walkers are non-interacting. They are not interacting with each other. And even in that case, it is not very difficult because they are non-interacting, whatever the joint probability distribution will be, it will like factor out, and it will be basically whatever was the probability, survival probability of the individual walker to the power whatever is the number of walkers. However, if we now make this lamp also moving around randomly, then it becomes a slightly difficult problem, because now each of these walker, I mean, you have to see that they are not independent anymore. The lamp is moving, so the dynamics becomes correlated, and it becomes basically difficult to approach this problem analytically. Okay, and it turns out that this problem has a very nice mapping in an important question, like to an important question in cell biology, which I will discuss. And let me give you like a quick introduction to this problem. So what you see is a spindle. So one of the important functions of a living cell is to distribute or segregate chromosomes in the daughter cells. And the mother cell does this by forming this very complex architecture, which is called the spindle. So it is made up of microtubules and other filaments, motors. And what it does, it forms this structure, like this bridge-like structure, which is tied at both the ends of the cell. And once this structure has been formed and all the chromosomes has been positioned on the equatorial plane, then this spindle actually pulls apart these chromosomes, as you can see, and then the following step is the cell division, and then it is distributed equally in the two daughter cells. So this problem itself, like this whole problem of spindle morphogenesis, that how this collective self-organization of the spindle takes place, is a very interesting question, and which is also very difficult and a lot of people study. What we are going to do is going to go a step back and see at one stage of the spindle formation is, if you think of this like the spindle, so what are the ingredients in the spindle? The constituents are the chromosomes, which has these small dots where, which are called kinetochores, and there are these two big dots from where the microtubule nucleates. These are called the spindle pole body. Now, oops. Yeah. So correlations in the sense that you have a searcher which is trying to find the lamb. If you construct a relative coordinate, it is x searcher 1 minus x lamb. Now if you take the second walker trying to find the lamb, that is x2 minus x lamb. So these two variables are correlated with respect to the moving lamb in that sense. Okay, so one important part of this spindle formation is initially at an earlier stage when these microtubules nucleate from the spindle pole body, and this chromosome, or I will now start calling it as a kinetochore because the microtubule attaches to the kinetochore, which is this protein complex on the body of the chromosome. So the one important question is how the microtubules attaches to the chromosome. Now you can think that you have some fluid and the chromosome is diffusing in the fluid, and this, once it is within the close proximity of the microtubule, there is an attachment. So you can clearly see what I was discussing with this random walker, many random walkers walking and the target is also moving, it actually maps to this problem. So you can think of many microtubules nucleating from this spindle pole body, and one of them actually captures that kinetochore, and the process is over. So this is what the mapping is, and we are going to try to understand with some whatever we know from this theory of first species processes. So as I have said, like, what is the mechanism of these microtubules attaching to a kinetochore? And it seems there are several mechanisms which exist, and I will just discuss one of the very well-known mechanisms, and there are many which is seen in different cell types, but this was, I think, like mostly studied, and this is called dynamic instability based search and capture. Anyways, I will just try to give you like a very coarse-grained view. There are more details in this process, but the main idea is you think that there is this centrosome from where this microtubule nucleates. This microtubule growth process is a stochastic process. So there are tubulins which can stochastically add, and then the microtubule grows, and there can be a switch from this growth to a shrink stage, and then the microtubule will start shrinking. And once it shrinks, it can completely shrink or it can rescue, it depends. But if it shrinks completely back here, it can again grow in a different directions. And then there are many microtubules which can do this thing. Okay, and meanwhile, this chromosome is also, this kinetochore is also diffusing, and then any one of the microtubules will capture this. So this has been studied before, and just let me tell you, so there has been a lot of theoretical work done around this problem. So already I think you have heard a talk by Raja Paul last week, who also works on this kinetochore capture. So Debashish has also worked like a lot on these details of kinetochore microtubule attachment processes. So this field is like, there has been significant contribution from the theoretical side. Okay. Do we know about any other mechanism? And it turns out, and this was very interesting, so this is in fission yeast. So in fission yeast, what was seen that this is how the experiment was done. In the experiment, what they did, they allowed the spindle to form, and then they suddenly like dropped the temperature to like a very low temperature, and when all these things depolymerized, and then they again brought it back to some higher temperature, and basically they wanted to see the whole process of capture happening under the microscope. And what they found was that, like, if you think of dynamic instability, this is how it happens. So there is a stochastic growth and shrinkage, and it keeps on happening with time. However, what was found in fissionist is the following, that the length of the microtubule goes, and then there is a pause for a significant time. So the length remains constant. And then it can shrink. And these time scales were measured, and while it is in this pause state, when the length is constant, it is getting hit by the background fluid molecule. So so in fissionist, this whole capture process happens inside the nucleus. So during this process, it is like a rod, and this rod is being pivoted at one of the spindle pole bodies, and it performs a rotational Brownian motion. And the kinetochore is also diffusing in the fluid, and this rotational diffusion enhances the chances of capture. So there were experiments, and this data was quantified carefully. So what was done, that you take a reference angle of the rod, and you measure the angle evolution with time, and you also measure the position of the kinetochore evolving with time, and you calculate the mean square displacement of both of these. And what was found, that the mean square displacement, the mean square angular displacement was also linear, and the mean square displacement for the kinetochore was also linear. So one can make an assumption that this is a Brownian process. So one can think of modeling this as like the evolution of the tip of a rod pivoted at one end, and then there is a target which is moving, and whenever they come in close proximity, there is a capture. And I will discuss the model in a few slides, but just I wanted to emphasize that there were some comparison of, this is called the fraction of lost kinetochores, which is nothing but the survival probability. So these dots are the experimental data, and these lines are from simulation, and it seemed that in a range of 3 to 5 microtubules is good enough to give you like a good comparison. And in this case, there were no fit parameters. All the parameters were independently measured across experiments. And what was seen, so this was done in room temperature. If you change the temperature, which means the diffusivity changes, and you still see without changing any parameter apart from the diffusion coefficient, which was measured from the experiment, you still see like a good agreement. Okay, so now the question is, we want to pose a hypothetical question now. So the hypothetical question is, so this is a mechanism which is very simple, unlike this dynamic instability, which requires like a pool of tubulin and all. So this is just pure like thermally driven rotational diffusion. So the question is, can we compare these two scenarios in a toy model setting? We can write down the models for these two cases, have the parameters measured in experiment, and compare by varying the microtubule number that which one is doing better. Okay, so the question is whether to pivot or not to pivot. And to do that, we can go and study the capture time statistics and like look into the problem in more details and characterize all the distributions and all. So, and the main idea is to compare the efficiency or the time efficiency, which one is faster as a function of microtubule number. Okay. So this is the work, all the work is done by Indrani, who is a joint PhD student of my colleague Dibyendu and me, and right now she is a postdoc at Ohio. So, I mean, most of the work I will discuss is from these two papers. And let me then introduce the models.
Amitabha, can I ask one question?
Yeah.
Yeah. So you are talking about the fissionist, right?
Yeah.
So in the experiment, of course, for fissionist, as far as I know, that it's not one microtubule capture. It requires, I think, 3 or 4 such microtubules have to be captured, right? So in the experiment, they end when 4 are captured or 1 is captured.
In the in the experiment. The kinetochore, you mean, not the microtubule.
Yeah. So microtubule, the kinetochore, each kinetochore.
One is captured.
Yeah, but actually in their fully formed spindle there.
So I think there are 3.
3 or 4, yeah, right.
3 to 4, yeah. So what I understand is, so this is basically the data is how the experiment is done is you have many cells like an array of cells, and you are observing basically the capture of, I think, one kinetochore, where...
See, in budding it is simple, only one.
Yeah.
But in fission is, so that's why experiment, how do they know that one has got attached or two had got attached?
No, see, this, I don't have a very clear answer to that. So what I know is they are looking at actually the capture of, like, so this is basically the fluorescence of the kinetochore complex.
Right.
So once this is captured, so this is considered as a capture event.
So does it have that kind of resolution that if there are 4 to be captured, it can distinguish first, second, third, fourth, like that?
And I don't know.
Okay.
Yeah, so this I don't have a very clear answer. So let me discuss the model a little bit. The model is very simple, and the idea is you have a rod which is pivoted at one end and it is diffusing, and there is a boundary which is basically the radius of the nucleus. Okay, and the thing is, you put the diffusion coefficients which are measured from the mean square angular displacement, and that will give you the dynamics of the rod. Now you can think of, so I have not wrote down the details, so you can think of deriving this equation of motion for this rod is like, you can think that you have a sphere, you have a point on the surface of a sphere, which is a Brownian particle, and it has this x, y, z, and each of them is doing a random walk. And now if you make a coordinate transformation in spherical polar coordinates, and because the length is fixed, you have no art dynamics, you will get a theta and phi dynamics. Okay, and when you do the coordinate transformation, you get all this multiplicative noise, and then there are these issues of how you interpret like Ito or Satanovich. I'm not going into the details right now, but if you want to ask, we can discuss later. So this is the equation, time evolution equation for the microtubule. And the microtubule is considered because typically the average length of the microtubule was seen as 1.5 micron, which is pretty below the persistence length. So it is considered as a stiff rod. The kinetochore dynamics is simple. It is like a Brownian particle evolving in 3D, and the diffusion coefficient is known from experiment. Now, for the hypothetical situation where we create a search and capture, we say that, okay, within the same boundary conditions, we have a growth rate of the microtubule. There is a shrink rate, growth to shrink, and then there is shrink to growth, and these are the rates which can be estimated from the experimental measurements. And this remains unchanged. And now we will study the capture in both the scenarios and try to compare. So this idea is clear to everyone? Huh. Right. In the previous one, yes. So that is a good question. So experimentally, what was measured was like a projection. Okay. And what you have is basically like only a polar angle. But then we did a calculation and see that if we make a projection of the actual 3D geometry, how much information loss is there. And that was like kind of compared, and then we moved to the 3D model. Yeah. Okay, so now we are comparing between these two. Okay, so I will not go into these details. And what can we compare? We can compare like the standard thing which everybody compares, the mean time of capture. So we can run these simulations like large number of times. We can calculate the mean time and compare between these two scenarios with varying microtubule number. So again, so this is a very toy model kind of setting. So these models are quite coarse grained, but our main motivation is some of these things which we are going to do, I mean, it will capture some of the key features. So we will not be able to compare really numbers like in a direct way. So this is the comparison we are making. Okay, in one case, the length is constant, doing a rotational diffusion motion. In the other case, there is a dynamic instability. Okay, so what do we see? And so this one is due to dynamic instability, this one is due to rotational motion. So they are very close. Probably the rotational motion is doing slightly better because the lesser the time is, it is better. And now as we increase the number of microtubules, also I wanted to show that this is basically the mean time which is measured from the experimental data. So from the experiment, they have a survival probability curve. If we sum over all those measurements, so this falls around this place, which also agrees that it is roughly like probably 5 microtubules. As we keep on increasing the microtubule number, we see that there is a saturation, and this saturation is not surprising because what is happening here, this saturation is the mean time of capture, which is dictated by the average length divided by the growth velocity. So you cannot, beyond a point, do better than like, because the microtubules in the dynamic instability model starts from roughly 0, and the kinetochores were at the center of the this thing of the sphere, and the average microtubule length is roughly 1.5 micron, and the diameter is 3 micron. So it is limited by like the average behavior. Now this is what we get. So they are roughly comparable. Maybe the rotational diffusion is doing better when we have like less number of microtubules. When you have large number of microtubules, I mean, it is saturating, so we cannot say much. But we want to ask a different question now.
Amitabha, I'm confused. So can you go back? Yeah, so in the dynamic instability, if it is not rescued, it disappears. So again, you start with a new one? So N is on the average fixed, or what is it?
No, so the thing is, we have the average growth rates, the growth rates, the shrink rates, and the switching rates, which we have.
I understand. What I'm saying that you have N microtubule number, right? So that is the initial number, I guess.
So we say yes. So what we do is we start with N microtubules.
Yes.
Suppose one of them shrinks, we immediately start another one. So the N is at a given time is always fixed.
Yeah, okay, so if one disappears, then you immediately start another one.
In a different direction.
In a different direction.
In a different direction.
Okay, and in the rotational diffusion, that number remains fixed throughout.
Yeah.
But they don't hit each other.
Yeah, that's a very good point. So in this model, so when we have less number of microtubules, it doesn't matter, right? But when we start increasing the microtubule number, one in principle should take into account steric hindrance or like this excluded volume interaction. But in this setting, we have not taken it. And you are right that as we start going to the limit of very large microtubules, probably they will play a role. So there this thing will become questionable. But now I will show in the coming slides that what could be like the difference it can make and why it will still give us like a comparison and give us like some essential understanding of physics.
And any number beyond, I think, 20 or so is in an unrealistic region, right? Because even in the mammalian cells, each kinetochore can capture, if I'm not wrong, biologists can correct me, I think 20 or 30 or something like that, and 100 is too large compared to the real ones.
The thing is, in this, one of these PNAS papers, early papers, I don't recall. So there they have considered like quite a large number of microtubules, right? So I don't recall the...
No, but you're talking about each kinetochore.
Each kinetochore. No, but this is one thing which we can discuss this later. So whether like when the kinetochore capture happens, whether basically it is like a single complex where you, instead of 3 kinetochores, you are capturing 1 kinetochore, or each of these 4 kinetochores has to be captured. Basically, whether we are doing a simulation per kinetochore or it is actually only 1 kinetochore. So you are right. So maybe one should not go to very large numbers.
Yeah.
Okay. So now, the question is mean time a robust measure to study capture in a confinement? And what we can do is, we can see this, suppose we study some capture event. So the capture time is T1. And we study the same capture event, the capture time is T2. Now we can compute something like the uniformity index, which is basically one of the times divided by the sum of the times. Okay. Now what we see that if this number is close to 1, it means that, like say, T1 is much greater than T2, which means that this T2 is negligible. Or you can have this omega very close to 0, which means T1 is much less than T2. However, if this omega is half, which means most of the stochastic trajectories are kind of close to each other. So what does it mean? If you have in a system very large trajectory fluctuations, then you will get a deviation. If you plot the histogram of this omega, you will see a deviation of the this uniformity index. So in that case, the mean is questionable because you have one trajectory which was very short, another trajectory which was very long. The mean doesn't make sense. Okay, so one should actually quantify whether this p omega for these simulations, whether it is unimodal or a deviation from unimodal. So if you see that it is non-unimodal, we should take it with a pinch of salt. So this idea is clear, right? And what we see that if we look at this rotational diffusion case, it is always non-unimodal. It is like for whatever number of microtubules we go. However, for the other case, the search and capture, as we go to larger number of microtubules, it starts getting like more of like a unimodal case. So the point is, mean time a robust measure to study first passage under confinement? Not always. So one has to be careful and see if there are trajectory fluctuations which are introduced. And in this case, because it is confined, you can have a scenario because the boundary also has an effect on this whole dynamics. Now the question is, if you mean is not reliable, then what else can you study? So are there any other time scales which you can characterize, which is independent of any kind of like initial conditions or anything? And this is where I want to, like, this is the important part of this talk. So again, you recall that if I have this kind of a capture process in free space, we know that the survival probability, as I was telling, it scales as a power law. Okay, depending on the dimension. Now, this is the simplest case, which means if I calculate the mean time, which is an integration of the whole survival probability, it can be infinite. So there are possibilities that the particle escapes to infinity because I am in open space. However, if I put a boundary, the capture is always bound to happen, which means mean time is always finite, and at large times, it will be exponentially fast. At very large times, this mean survival probability will go exponentially fast, and then there is a time scale tau, which is basically a characteristic time, and it tells you like the capture time for rare capture events. It is corresponds to like very low survival probability, but it is something which is independent of, it is only dependent on the system parameters. So it gives you an upper bound to the capture time scale. So this is something I quickly wanted to tell you. So I have talked about this. So if you have a static target with n independent searchers, your survival probability becomes like factors out, and you have the survival probability to the power n. In confinement, you always have an exponential tail, which means when you do this to the power n, your characteristic time is nothing but whatever the time you have calculated for a single worker divided by n. Okay. The problem is when you have a moving target, nothing can be solved analytically except for the simple case where the target and the searcher are diffusing with equal diffusion coefficient. And anything beyond that for n greater than 1 and even at higher dimensions, so we cannot do anything analytically, so the problem has to be studied numerically. And now the question is, this tau, this corresponds to a very low occurrence probability, so how you are going to estimate this tau? Okay, I will skip this. These are the results in open space which has been done earlier. I think now it is not relevant. So, like some of the analytical results. So the question is how to numerically estimate this characteristic time. So again, what is important is this exponential tail, and let me try to give you like an idea. So suppose you are measuring the survival probability, and the survival probability can have two scales. So this survival probability is plotted in a semi-log scale, which will have this kind of, if it has an exponential part, it will have a linear decay. Now, if it has multiple time scales, it is possible that what you will be estimating as a typical time is actually the first part of the survival probability, and it does not capture the actual typical time. So you have to really go up to very small survival probability values to get an idea of this typical time scale. Now, the problem is the following. So you see, you are doing it numerically. It's a very challenging task. Suppose you start with 1,000 different random trials, which you are running the simulation, and out of this 1,000, every time something gets captured, your survival probability is going low and low. By the time you have reached here, there are only few left, so the fluctuations are also very large. Okay, so numerically, you will not be able to really reliably estimate this time scale. So how to do that? So you need a very good precision, and the way to do that, and that is what we try to propose here, and this is something like using a replication, and this was like the ideas are taken mainly like this was proposed earlier by Grasberger and Nadler and Meher and Grasberger way back in 2002. So the idea is the following. Suppose you are looking at the capture process, so each of these is a cell, and this microtubule will bind to capture the kinetochore. So you start with some configuration and the survival probability is 1. Now, out of these, 2 of them gets captured, which means now the survival probability is half because 2 has got captured. What you do now, because your ensemble size has reduced, what you do, you replace these 2 which were captured and just replicate which are 2 remaining. As a result, your survival probability has not changed, but what you are starting is whatever was the survival probability here, and then from there you have increased the number of ensemble, and again when it further goes to the stage that few more are captured, you keep on replicating and removing and replicating with the existing ones. And if you keep on doing that, your survival probability can go up to an order of like of the order of 1 by 2 to the power n. You can go to up to very small scales, and this is what I wanted to show you, and this is the important part. So this is basically the survival probability for the rotational diffusion problem with 3 different initial conditions. In one case, the kinetochore was very close to the microtubules. In one case, they are far away, and in one case, they are somewhere in between. Now, you see, this one is the one where they are very close, because as you start the simulation, some of them gets immediately captured. Okay, then there is a huge drop. But then this whole thing evolves slowly. So, if you want to calculate the mean time, you have to integrate the full curve, which means you are taking information from here and here also, which means you also have an effect of the initial conditions. However, if you calculate the typical time, you are only looking at the tail, the slope at the tail, which are independent of what is happening earlier. And they are like unaffected by whatever was the initial condition dependence. So that's why it is a very robust measure. And now we can say that because there is an ambiguity with the mean time, we can compare the same 2 scenarios like rotational diffusion versus search and capture by computing the characteristic time, which is independent of all initial conditions. So I also wanted to show you that we also studied the corresponding non-biology like a prototypical model where you have a lot of point particles and target, and the idea is to study the same thing and look at like how the typical time varies as a function of the number of searchers. Okay, so let us again compare the same two scenarios, but now with this new time scale. So what we see here is for the typical time, as we change the microtubule number, the case of search and capture keeps on decreasing, whereas the case of rotational diffusion actually saturates. Now, as I was saying that, so it seems that there is a role reversal. So initially, for small number of microtubules, the capture due to rotational diffusion was doing better, but as time progresses, as we increase the number of microtubules, this saturates and the other one takes over. Now, as I was saying, so this is the case of rotational diffusion. We have not taken excluded volume interaction into account, and that is an important aspect. But what we think that if you introduce excluded volume interaction, this saturation will happen even faster. So this will saturate much faster. So you will see this role reversal at a much earlier, smaller number of microtubules. So what we see here is, if we have less number of microtubules, rotational diffusion is efficient. If we have more microtubules, it seems that this one is efficient. So this is just a finding from this toy model. And now the question is whether it is true or not. And that has to be looked at. But an interesting thing is you see a saturation in this case with the rotational diffusion. So you might be intrigued that why there is a saturation in the time scale as we increase the number of microtubules. And the question is and the answer is the following. So what happens? This microtubule is pivoted and doing a rotational diffusion. Now when we increase lot of microtubules, each of them are doing a rotational diffusion, you are basically creating a surface. And at some point in the limit of n tends to infinity, this particle will just get captured on the surface. You cannot do better than that. Okay, and you can check this. How can we check this? We can create a toy model scenario where we have a circle, we put some dots along the radius which are diffusing, and we have a particle which is diffusing in the upper part of the circle. And now we keep on increasing the number of circles, and if we start doing that, it will saturate. Instead, now we can study an analytical model where we have an absorbing boundary. We have a particle diffusing on the other part, upper part of the circle, and if we put an absorbing boundary condition here in 2D, what we see is we get this dotted line. Basically, we get this value of tau. So it exactly saturates to this value. So it is limited by when we go to a large number of microtubules, we have this. So the other question is, we saw if we again look at the way this time scale for capture scales with the number of microtubules, it seems there is some kind of a power law exponent. Now, I showed you that if we have the scenario where we have a static target and n walkers trying to reach the target, the time scale is scaling as n inverse. Okay, and here we see when we make this target moving, there is a deviation from this exponent, the exponent is lower. Okay, now this is a difficult problem, but we wanted to see how this exponent looks like, so we wanted to study this in a prototypical model where how this exponent depends on the number of searchers and diffusivity of the search and target and the system size. So I will not go into the details, but it seems that it mainly depends on the ratio of the diffusivities. It is independent of anything else. If you just keep the ratio of the, change the ratio of the diffusivity, I mean, for a given ratio of diffusivity, which is non-zero. What I mean by diffusivity is diffusivity of the target by diffusivity of, say, the Walkers. So if dt is 0, it goes back to this for dt equals to 0. If dt is not equal to 0, then it gives a power law, and that power law depends on the value of the ratio, how fast you are making the target relative to the searchers. Okay. So now the point is, see, let me go back once again. So we are talking about some typical time scale, which is associated with very low occurrence probabilities. Now, an experimentalist will tell us, like, why is it even important? We cannot measure it in experiments. Like, if we were asked to measure a typical time, a characteristic time, maybe it is initial condition independent, it is not measurable. It is very difficult because we cannot go down to a survival probability which is very low. What is a way out? Like, can we estimate this in experiments? And here what we try to propose, and the idea is based on extreme value statistics, and it is the following. So suppose you are an experimentalist, you are measuring these capture times. You have like n cells, you are measuring like how a capture event occurs in each cell, and you record these values. Now I can ask, and each of these, they are independent realizations, so each of them are coming from some, these are independent like distributed random variables, identical and independent. Now I can ask, so, and each of them has some parent distribution which is f. Now I can ask, like, what is the maximum value? Okay, or what is the leading value of these time scales? And this is nothing, but if I want to ask that what will be the distribution of this leading time, the maximum time, this will be the cumulative probability of t tilde greater than t max, which means I have made this many measurements. So this t tilde has to be greater than each of these t1, t2 and tn. And because these are independent and identical, I can again factor it out, and I will have basically the cumulative distribution of this parent distribution up to t tilde to the power n1. Which means, and this, if you go to the literature of extreme value statistics, it is known that, whatever is the parent distribution, it eventually goes to, if you calculate the extreme value distribution, it goes to 3 universal parent distributions. And if, for example, your original distribution of the times were exponential, it goes to a Gumbel distribution. So this is known. So if you have an exponential, and we know that because the capture is happening in confinement, the large scale, like at asymptotic time scales, this survival probability is exponential, which means we know that this will be a Gumbel distribution, which has a tau here. And one can calculate the mean and the tau, so relate the tau in this distribution to the variance of these measurements. Which means what we can do now is the following. So we can do n measurements. Now we break it up into n1, n2. In this way, for the n1, we find out the maximum times. For the second row, we find out the maximum time. We have a set of times which are n2 number of times, and we calculate the variance. And that variance with this formula will be related to the typical time. And we can check that because we have that very highly precise method to compute the typical times using a replication algorithm, which is kind of exact. We compare for different number of samples how this agrees. We see that there is like some reasonable agreement where you have a large number of sampling, but when you go to very small number, there are deviations, but it still nevertheless gives you some idea of what would be the typical times. So let me conclude. And so we created like a hypothetical model, and tried to understand that whether like we can compare two different scenarios which has been seen inside cells for kinetochore capture. And what we found that this mean time can be misleading for studying first passage processes inside confinement, and one should use it with some caution. On the other hand, if there is a way to compute the characteristic time, it is which is associated with the rare capture events, this is independent of initial conditions, and I mean, it can be computed. And yeah, so I will stop here. I think I'm quite on time, and this work was done by Indrani, all the work, and she was a joint PhD student with me and Divendru. And yeah, and thank you for your attention.
Okay, time for questions?
Yeah.
Yeah. See, if I understand correctly in the first part, okay, so their actual thing to compare would be the solid angle, because each of these rotational diffusion ones, they are covering a solid angle, and so the time taken to capture that should be somehow comparable to the microtubules that you throw through dynamic instability in the same solid angle, right? Because they are non-interacting.
They are non-interacting.
Right. So that's why.
I don't know. So you probably missed this part, but just a second.
Yeah. When you are comparing this.
Yeah. So if you see this rotational diffusion, I don't know if.
Yeah. So my intuition would say that basically these two are identical except for the fact that if I have taken a microtubule and it is doing random rotational diffusion, so it is tracing some solid, covering a solid angle within a given time.
Right.
And within that time, which on how many microtubules doesn't matter, you send to probe that region, okay, they should be just comparable. It's just a question of how you cover a solid angle. By throwing many of them or by sweeping one single one, because they are non-interacting.
So the solid angle can be calculated because we know the diffusion coefficient. So one can find out like how much surface area, like the solid angle, it will probe out.
Probe. Right.
So what you are saying is, if I have that estimate, I can think that how many microtubules I should throw out.
In that, so then they should come out to be comparable, right? Because
but the problem is you cannot increase the efficiency with rotational diffusion beyond a certain point by increasing the microtubules, precisely because you see so this is the typical time, not the mean time. So this is what you see is a saturation.
Yeah, I understand. What I'm saying that just one microtubule is equivalent to many. One microtubule rotation is equivalent to many microtubule thrown within the same solid angle.
Possibly, yeah. So if I understand correctly, what you are saying is like whatever is the solid angle, like what will be the equivalent number of...
These two comparisons are not on the same footing. That's what I'm trying to say. Because one single microtubule rotational diffusion is equivalent to sending several microtubules to cover the same solid angle through dynamic instability.
Okay, so I think comparison has to be done different in what way? So here we are just taking the dynamics and we are saying that like it is like a numerical experiment we are doing. We are comparing the time.
So this is straightway comparison n versus n.
Right.
But what I'm saying is that the rotational diffusion actually is doing the job of many. Rotational diffusion by single one is doing the job of many microtubules which are doing the same job through dynamic instability.
True.
So when you compare them, so obviously it will appear, you're not comparing them on the same footing. That appears to me.
But if I think of just like how much time it takes for a capture, so I see that this thing is true when we have less number of microtubules, right? Like the analogy you are giving, if I understand correctly. I mean, beyond the point, this comparison is no longer like, I mean,
Yeah, probably because
this one
the
is showing
solid angles of different ones overlap, right, in the rotational one.
yes, so basically what happens is it is basically tracing out a surface, and by further increasing the microtubules, you cannot increase the capture efficiency because everything will get captured on that surface, basically. You cannot make it any more better.
Yeah, probably that's the.
So, but if I just think both of them as two mechanisms and I say that, okay, I want to see time-wise which one is efficient. So, I mean, I don't see why I cannot.
Intuitively, it appears to me that having fewer rotational ones is more efficient. It just if you want to hit a target,
Yeah.
a flying target,
Yeah, yeah.
You don't shoot too many missiles.
Yeah.
You know, so.
Yeah, that is what it looks like. Yeah. True. Yeah. But it is pivoted at one end. Huh?
So you showed this, the length is constant.
The length is constant, yeah. So the length, what we have taken is what is basically like the average length, which is seen in experiments. Now, see, if you have a rod, and if you are considering rotational diffusion, the diffusion coefficient is a function of length. It scales as 1 over L cube. So when you make it longer, the diffusion gets slower. So one can show that. So we have that factor also taken when we compute the diffusion coefficient. So it might not be that easy. So when you make the length of the rod longer, the sweep will become slower. It is a very strong dependence on it. It's 1 over L cube. Who? So in this scenario, it is a first passage process. So whenever the kinetochore hits the microtubule, the event is stopped.
So I mean, there are many microtubules, right? So I mean, if one is stopped, you have to consider it also. I mean, it will restrict the movement of the other microtubule.
Yeah. So in this simulation, we don't consider that. We are just considering as an event where this thing actually stops. And usually, I don't know, I mean, there are experts can tell. So usually when a capture event occurs, this chromosome is immediately brought back and positioned in the spindle, right? So we are taking like something like in that spirit that you have only a single capture event. Once it is captured, it is stopped. Yeah. Okay.
It is always our theorist thing is oversimplified.
Okay.
That if it goes there, it is immediately captured. So it is not necessary that immediately captured because there has to be binding that has to take place.
Exactly.
And even after that, if it is just from one side, okay, it's not enough because it has to be bipolar. So from the other side also, microtubule has to come and bind it. So this is just single capture. It doesn't mean that the kinetochore is positioned just by that. Right?
So there is like a second process which actually.
Because the two spindle poles. So from the other side also microtubule has to come and bind and then position it.
Oh, I have a question.
Can I ask another question?
Yeah.
Go ahead.
Yeah.
Actually, I have two questions, so maybe you better. Yeah, so in the model with the free random walkers and the random target, in your second model, you took point-wise searchers.
This one, right?
Yeah, so I mean,
Ah.
yeah, so like I don't know, since microtubule are of course linear, but you took point-wise searches, so
No. This is nothing to do with the microtubule problem. This is you can think of it
It's
as a
just
statistical
search-
mechanics problem where I have point particles non-interacting. I want to see that how within confinement they capture a moving target and as a function of different dimensions. So this problem per se doesn't have directly anything to do with the actual capture problem within the cell, but nevertheless what I was trying to say that some of the key features that you have a power law dependence of this time scale tau on n where the exponent is less than 1, it is seen in both the scenarios. So this is something which is common, and this happens because you have put movement of the target.
Okay. Second question is, I don't know if you were there, of course, Professor Raja Paul's talk.
Not there.
Okay. Then I don't know. Okay, then I'll ask this question later, maybe.
Okay.
One quick question. You showed this comparison between these two cases in which one, the microtubule size is constant. It searches by doing rotational diffusion. In the other, it's changing its length and going in different directions. But in simulations, did you mix these two methods together and see if the time it becomes faster?
Yeah, so we have done that. So then this saturation disappears. So then,
but is there there could be like some X percentage of this method along with 1 minus X of the other method which will minimize the time? Is there a minimization?
No, that we haven't done actually. What we did was a very simple-minded thing, like a full simulation where we keep in a model both rotational diffusion and dynamic instability, and the dynamic instability is as per whatever the rates were from the experiment, that initially they grow and stay, pause for a long time, and then they shrink. But what you are asking is how to
sort of phase space
optimize
scan.
phase
space scan, which may not be relevant for biology, but just
No,
generally, is there a minimum?
We didn't do that.
And the second question I have is that the rotational diffusion, the equation you showed and said that is it Itos or Stratanovich?
It is Stratanovich, so I will tell you. So this equation, actually, as I told that basically you write down and put a constraint that a point is diffusing on the surface of a sphere, okay, and do a coordinate transformation. So basically x equals to a sine theta cos phi So soon, like in the theta phi, and A is not there, A is fixed. So all the noises mix up, it becomes multiplicative. Now, because this is like, one can think that the noise source is because of the thermal background, it is like an extrinsic noise, and it is not related to the intrinsic nature of evolution. We consider it as a Stratanovich convention and convert it to a Ito differential equation, and that is what is written here.
Amitabha, the question that Debashish asked, the other Debashish asked you, is probably relevant also to compare with Nenad Pavin and Eva Tolich's model, because they had hinge and it was rotating at the tip.
This is the same model.
Yeah, but you have either full rotation or you have dynamic instability.
So this is full rotation within a confined.
Exactly. So what I'm saying, that if you combine the two, what Debashish was saying, and if I understand correctly, then there is a way of saying that what length remains fixed, and from there it hinges and searches.
No. You mean the first question, which
Yeah. First question, yeah.
Yeah. So the first question, if I understand correctly, what he's asking is
can you mix the two?
How can you mix the two?
Yeah.
What ratio of?
No. So one way of mixing would be that one single microtubule, you have to put a hinge somewhere, and around that hinge, part of the microtubule in the front, that sweeps, rotates. The other part remains fixed. Now the question is, where do you put the hinge?
Okay.
Okay, so this is like somewhat like, you know, what your dynamic instability does, but not at the tip, but somewhere in the middle. Right?
Yeah. Okay. Yeah. I mean, that's that's a nice way to look at it. Yeah. Right.
Yeah. Any more questions?